the neurobiology of love. So there's lots of reasons why we want to study love or affiliative behavior, as psychologists tend to talk about it. There's so much variation in the kinds of bonds that are formed, but those bonds are very, very strong. So we can go from very strong bonds, bonds that get broken, bonds that we're not sure what's going to happen. But we talk about this because if you look, this is a great example of how neurobiology relates to so many of the other topics that are taught at NYU. How many novels, how many plays have been written about love and those feelings, those deep feelings that we have? We all have them. What could be more interesting and more important than understanding the neurobiological basis of this? But wait a second, it's harder. It's not just like vision, where you have eyes and you have photons coming in, or just touch. This is a much deeper, higher cognitive function. How would you study this? Well, a guy named Harry Harlow was one of the early psychologists that started to try and systematically look at affiliative behaviors. And he identified 3 different kinds. Parent-child, obviously very strong bonds there. Filial, brother-sister, bonds there. And male-female, partner bonds, which this last one is the one that we're going to focus on today. And to start to ask what brain areas and what kind of brain chemicals might be involved, we jump back to the hypothalamus. Now, the hypothalamus, we'll remind ourselves, what is the hypothalamus good for? And I believe I told you the key for the hypothalamus, the 3 F's. Feeding, fighting, and making love. 3 F's. Okay, so we talked, and for the third F, we're going to focus on 2 nuclei that we talked about before, so you're very familiar with them. 2 key nuclei in the hypothalamus, the supraoptic nucleus and the paraventricular nucleus. Okay, they're up here in the hypothalamus, and they connect to the axons in these 2 nuclei, go down and terminate where in the posterior pituitary gland. And in the posterior pituitary gland, they secrete one of 2 key neurotransmitters that will be key for the rest of this lecture, vasopressin and oxytocin. Why are we starting here? We're starting here because both of these neurotransmitters have been strongly implicated in higher social behaviors, social interactions, and in particular, territorial aggression. And so that's why people were particularly interested in vasopressin and oxytocin for this related form of kind of social behavior, which is male-female bonding. Now, we need a little bit more than just a brain area and 2 of these hormones to study. We need a system to study. We're going to study in humans. There's certainly lots of variability there. Well, it's hard to really get into their brains and really figure out what happens. Instead, as I mentioned last time, we are going to study male-female partner bonds in an animal model system that seems to be made for this study. And that system is the Vole system. Voles are cute little rodent animals. Look at him, he's very, very cute. And it turns out there's 2 different species of voles. The first species of vole is called the prairie vole. And these all live in the Midwest, out in areas like Montana. They dig underground. So here what I'm showing you is the little burrow pathways of the cute little vole. Prairie voles are very clearly monogamous. They form lifelong pair bonds between males and females, and we'll talk about exactly how that happens. They live in multi-generational family groups. They are the family value voles of the Midwest, okay? These are the prairie voles. Males and female form breeding pairs and are always very often in close proximity to each other. So if they are pair bonded, they just spend a lot of time near each other. These are the prairie voles. parent-child, both parents take care of the young. It's not just the mothers that take care of the young, but because they live in these family units, it's both the mother and the father that are taking care of the young. Now, beware, this is a kind of a dramatic picture. Intruders of either sex are rejected by both sexes. So in this multi-generational family unit, this could be a male or a female attacking prairie vole, very vicious, and attacking intruders of other that are not part of the family. Now, in contrast to the prairie vole, who is monogamous. I was going to say very monogamous. I guess you can't be very monogamous. You're just monogamous. There is two related species. I'll focus on the montane vole. So the prairie vole versus the montane vole. Montane voles, in contrast to the family value prairie voles, they are the partying voles. They are promiscuous. They go with any vole that comes along. They live in isolation. They're just not that social. They're happy to be alone. They have so low social contact. Of course, there is mating and they survive as a species, but males show little or no parental care, unlike the prairie voles. There is also a very relatively short time that the mothers are taking care of the infants. So those prairie voles, those montane voles, early after they grow up, they're kicked out of the nest and they have to, fend for themselves. So very, very different kinds of behavior. So now we have prairie voles and montane voles. And I can tell you a little bit more about the just behavioral patterns, particularly of the prairie voles. How did these lifelong pair bonds happen? Well, it's actually very interesting. So there is these little prairie voles are living in their family units, and at some point, the female voles in the family come to an age where they are sexually mature, but they're still living in their family. And what happens is, when this female prairie vole is exposed to the urine of a non-family member, or sniffs the urine of a non-family male member, that's it. True love. The scent of the urine of a male in the other family will initiate pair bond formation. What do I mean by initiate pair bond formation? I mean initiating intercourse for a long time. What do I mean by a long time? 30 to 40 hours. So at pair bond initiation, these little prairie voles are going at it for 30 to 40 hours. Okay, they come up for air, for water, maybe a little food, smoke a cigarette, I don't know. But 30 to 40 hours, that's amazing, right? And so during this time, we know that these key neurotransmitters, or these key hormones, oxytocin and vasopressin, are surging in both animals. So we know that something is going on. But after this 30 to 40 hours of intense interaction, there is a lifelong pair bond formed. Okay, so let's just go over the differences between prairie voles and montane voles. The mating system, prairie voles, as I said, monogamous, starts out with a bang, 30, 40 hours of intercourse. Montane voles, promiscuous, dime a dozen. Parental care, biparental in prairie voles, maternal and montane voles, and short maternal, not very long care of the montane mommies for the babies. Partner preference is very, very high. It is clear that the two animals in the pair bond, they spend lots of time together. Montane voles, if you've mated with somebody, doesn't matter. Selective aggression, prairie voles high, selective aggression against other people, and montane voles are much more, low-key. Social contact high overall with their family members in particular. Montane voles, again, are more isolated, and they'll come together for their promiscuous kind of mating. Okay, so this is, once we realize this, thought this is great, okay? So now we can look at what's different in their brain and possibly look at the role of oxytocin and vasopressin and underline this dramatic difference between these two vole species. Okay, so affiliative behavior in prairie voles and montane voles. Well, now the problem is you can't go out and study their brains when they're running around in their family units in Montana. You actually have to bring them into the lab. And so what they next develop is a series of tests to be able to quantify their affiliative behaviors, and particularly their pair bond formation. So then you can go and look in at the effects of changing oxytocin, giving more oxytocin, giving less oxytocin, and screwing around with their oxytocin and vasopressin to see what the heck it's doing in either the montane vole or the prairie vole. So here are two tests that are used. So this is a female choice test, a female partner choice test. And here is the lucky female. She is not tethered. She can walk around wherever she likes. And she has a isolation chamber here, or she can walk through the hallway and spend time with a male stranger. She doesn't know this male, and he's tethered to the wall so he can't follow her or anything. And this is her partner, so she's pair bonded with the partner. And you can just look to see how much time she spends, and if this is a female prairie vole, you know that she's going to spend significantly more time with her partner, very little time alone, and very little time with the male stranger. Okay, so this is what we do. We can test using this partner preference test, and we can compare the difference between montane voles and prairie voles to see whether they exhibit the same kind of very dichotomous patterns in the lab as were observed, as the ethologists observed in the wild. And this is what they see. Here's what they see. They mate the pairs at the time for 24 hours. So they don't give them quite as much time as they have in the wild, but this still forms a good pair bond. And here is the female that has been mated, and here is the partner that's tethered. You can see by this line here, the partner that's tethered and the stranger that's tethered. And let's look at the amount of time that this female prairie vole or montane vole spends with the partner and the stranger. Prairie vole spends the majority of the time in minutes over 3 hours in the partner's cage, a little bit in the neutral cage all alone, and a little bit in the stranger's cage. Montane vole, look how different this is. Montane vole is like, I want to be alone. Just leave me alone. Totally a lot of time in the neutral cage, not so interested in either the partner, and for the montane vole, this is the, by partner they just mean the male that she was mated with, or the stranger. Okay, so this is great. This means that we can see the same very dramatic differences in their partner behaviors in the lab. Now we're going to look at another test, which is aggression. So aggression can also be studied, and I said that was very different. There's a lot of aggression in the prairie voles that are protecting their partner, they're protecting their family, and the montane voles that say, whatever, live and let live. So here's how this test works. This test, while the other test was done on females, the partner test is done on males. And so first, before mating, you can put two males together, okay? And one of these males, the open circle male, is then mated for 24 hours, either prairie or montane vole. And then this same male, this open circle male, is now put in with a different male. This male here is different from that male there. And we can look at the difference and see and make sure that the males are showing kind of species-specific reactions, and we absolutely say that. Prairie voles, they show a lot of aggression, particularly after they've mated, and we can see before mating, frequency of aggression to this other guy before mating, it's like, he's happy with the guy, but after mating, you see huge increases in aggression, and then after long-term housing with the female, it's just another control, you also see aggression. Montane voles, doesn't matter if there's been a mating or not, before or after, there's equal amounts of aggression. So this is great. This means that we have an assay that works in the lab that can demonstrate these species-specific differences. And in particular, we're going to focus on these striking behaviors that the prairie voles are showing for their affiliative behavior. So before we go into manipulations, given that oxytocin and vasopressin were key targets of interest in this higher level social interaction, social pair bonding, you can ask, well, the question they asked was, where are the receptors for oxytocin and vasopressin. So I'm just going to tell you that both the prairie voles and the montane voles synthesize oxytocin and vasopressin, and it's secreted out of the posterior pituitary. But the major difference is where the receptors are in the brain. Okay, so let's look at that. Here are the receptors for the prairie voles on the first row and the montane voles on the second row. Here we're looking at oxytocin receptors, and here are vasopressin receptors. And the only thing I want you to appreciate from this slide, you don't have to know what these different abbreviations are. All you need to know is that the oxytocin and vasopressin receptors are in different places, different brain areas in prairie voles and montane voles. Okay? That is the key take-home from this slide. Now, you tell me, what would that mean? What does it mean that you have receptors for oxytocin and vasopressin in different brain structures? What would be the implication of that if that's all you knew about prairie voles and montane voles? I mean, you also know that they have very different behaviors. What would be the implication of that, do you think? Yes?
The different voles would react differently to the same...
Exactly. The different voles would react differently to oxytocin and vasopressin because it's influencing different brain structures. So when oxytocin and vasopressin are released, it's simply not doing the same thing. Okay? Does everybody get that? Any other ideas about what the location of the receptor might tell us about the role of oxytocin and vasopressin? So the identity of the structure will be very, very important. If all the oxytocin and vasopressin receptors are in visual cortex in one species and in somatosensory cortex in the other species, that would have very different implications. One would be, perhaps visual cortex is obviously visual cortex would be highly influenced by these hormones, and other brain areas would. But the function of these brain areas really tell you a lot about what these structures might do, what these hormones might do. So now we get to a key question. What is the role of vasopressin and oxytocin in pair bond behavior? Okay, we know it's been implicated. We know it's in the prairie vole and the montane vole. Now this main question, we're going to focus on the prairie vole. The prairie vole has this active behavior of pair bond formation. We want to ask, what is the role of oxytocin and vasopressin in the female brain? And what is the role of oxytocin and vasopressin in the male brain for either female preference or male aggression? Okay, so let's take them one at a time. Okay, oxytocin in the story. Let's take females first. We know that oxytocin surges in females after mating, not just after that first big bout of mating when the pair bond is formed, but after every mating situation. And we know that pair bonds form after the first mating. And so the question is, could oxytocin be related to this pair bond formation, which is I'm calling love here? So what is the test that we're going to do? We are going to do the female choice test. So female prairie vole here with a male stranger prairie vole and a male partner prairie vole. And first, we're going to look at an unmated female. Okay? Unmated females given cerebral spinal fluid. So this is just normal behavior. This is just to show you what the female will do. Not surprisingly, this is unmated to recently unmated. Still has a big preference for the partner, little bit of preference for neutral, and little bit of preference for stranger, but still has a big preference for the partner. Now what we're going to do to the female, again, not recently mated, we're going to give them either oxytocin or vasopressin. OT is oxytocin and vasopressin is AVP. Okay, so let's look at vasopressin. Vasopressin has similar kinds of proportions. I can tell you that statistically, this graph is not different from this graph. So let me just make sure that you know how to read this graph. On the y-axis is percent of time with a preference, that is percent of time spent with either the partner, the neutral, or the stranger. So the bigger the bar, the more time she spends with the partner, the neutral, or the stranger. So I'm telling you that when they inject vasopressin, these bar proportions are no different from these bar proportions. There's a little variability, but still. The big difference, the significant difference comes when in this unmated partner, hasn't been mated for a while, you give her a little squirt of oxytocin in the ventricles. And now her preference for the partner really goes up. It actually mimics mating. So after mating, the females tend to have an even stronger attachment to the partners, and you see the significant increase in the amount of time the unmated female spends with the partner compared to a complete stranger. Now we're going to do the key experiment. So this confirms that oxytocin. This suggests that oxytocin without mating can actually mimic mating. So, and we know that's consistent with the fact that after mating, oxytocin is released. So we've confirmed that when you simply give oxytocin, you form a bigger pair bond with a partner that hasn't been mated with recently. Now can we switch that the other way? Let's look at antagonists of both oxytocin and vasopressin. We're going to look at antagonists in females that have been mated. So now the female that has been mated has a huge preference for her partner. Why? Because she's just had a surge of oxytocin, and that surge of oxytocin, based on the brain areas that it's being affected in, causes a much closer proximity of the female to her partner, and spends just a little bit of time with a stranger and no time all alone. Now what we're going to give this female who has been mated, this is her normal behavior, just glommed on to her partner. We're either going to give her an oxytocin receptor antagonist. What does an oxytocin receptor antagonist do? Yes? Blocks the release of oxytocin. Close, but no, yes?
It blocks the receptors of oxytocin?
It blocks the receptors of oxytocin, yes. So oxytocin receptor antagonists are blocking oxytocin receptors, okay? And then we're going to give either an oxytocin receptor antagonist or a vasopressin receptor antagonist. Again, blocking vasopressin receptors. So look what happens. Let's start with vasopressin receptor antagonists. Here, what happens is there's no change in the proportion of time that the animal spends with the partner. Still very, very high. Now this female appears to be spending more time alone than with the stranger, but the main finding is that it's not changed at all, this glomming on that the female prairie vole has to her partner. Let's compare this graph with now this one. Now what we're doing is squirting in oxytocin receptor antagonist to the ventricles, and now we've brought back down the amount of time this mated female spends with the partner back down to pretty close to when she hasn't been mated at all and to normal. So, but she has been mated. So blocking oxytocin receptors takes away this partner behavior, this affiliative behavior in the female. So what this suggests is oxytocin is necessary to be able to express in females this partner preference after mating. And it could also, it's also sufficient to, when you have an unmated female, to actually mimic, to cause a mimicking of the high partner preference, even a female that hasn't been mated. Vasopressin does nothing in the female. In the female, it's all about oxytocin. And we say that oxytocin is both necessary and sufficient for the expression of female partner preference. Okay? So that's an important phrase to understand. These experiments show that oxytocin is both necessary. You absolutely need it because if you block it, you don't get it, and it's sufficient. If you give it and it's, the female isn't expressing the behavior and you give it, it's sufficient to actually allow the animal to express that behavior. So oxytocin in these female prairie voles is very, very powerful. Now, what about the males? In males, the story is a little bit different. In males, it's not oxytocin, but it's vasopressin that surges in males after cohabitation. We know pair bonds form after cohabitation, and so we ask, just like we ask about oxytocin in the females, in the males we ask, is vasopressin related to this pair bond formation? And so here, we're not going to do the aggression. We're still going to do the same partner preference, and I can show you what those results are. Males that have been not mated recently, they can go with their partner or a stranger. And males have a little bit, they spend some time with the partner, some time with strangers, and some time with neutral, but they don't have, when they've been unmated, males are a little bit different from females. They express similar spending time behavior with both a stranger and a partner. Now what we're going to do is in this unmated male, we're going to give oxytocin. Here, this graph is no different from this. Again, the male is spending about as much time with the partner as the stranger. But when we give AVP, which is straight vasopressin, in the ventricles, now the male starts spending much more time with the partner. Okay, now we're going to do the same experiment. Now the male, instead of being unmated, the male is mated very recently with the partner and then gets put in the same little box situation. So it's a male here now, and there's a female that's tethered. That's the partner, and a female, partner female is here, and stranger female is there. And what we see is once the male has mated, now you see 100% preference for the partner. Now the male doesn't leave her side. Okay, so the mating in males may be a little bit different. It may not spend quite as much time unmated, but mated, no problem. The gloms onto that female. Now let's look to see when we give oxytocin receptor antagonist or vasopressin receptor antagonist. Let's look at the oxytocin receptor antagonist first. And what this does is absolutely nothing. It does not change his behavior at all. So even a lot of oxytocin receptor antagonist is not changing his normal behavior to have a high preference for the partner after he has been mated. Now, let me just make sure you understand CSF is, remember, cerebral spinal fluid. It's normal kind of plasma for the brain, and this is a control. We're injecting something that should do nothing to the brain. Yes?
Can you clarify, what do you mean by unmated but it also has a partner?
Yeah, unmated means recently unmated. So he hasn't mated with that partner in a little while. Okay, but he's formed a pair bond. Okay. So it is a partner. That's a really good question. And the same thing, same definition goes for the females. He's a partner, but the female hasn't mated with that partner for a while. Okay, so something special happens even after the pair bond formation happens after even the partners mate. And what happens is surges of oxytocin or vasopressin come up, and that causes even more partner preference. Okay, so that's what we're seeing here. Good question. So males have mated with their partner, and they now have revivified this preference for the partner. Giving oxytocin receptor antagonist does nothing. And now we're going to look at giving vasopressin receptor antagonist. And here we see a huge decrease by about 50% of the preference for the partner. And this goes down to, not so different from a preference for the partner when he's unmated. So here the finding is very different from the females. In males, the finding is vasopressin is both necessary and sufficient to produce this strong partner preference behavior in the male prairie voles. So, yes? Okay. So the answer here, I love describing these experiments because it's a very, very complex kind of interaction, social pair bonding. But at least on the surface, you have a clear result with oxytocin really underlying one key aspect of pair bond formation in the females, and vasopressin underlying the same behavior in the males. Okay, so what about humans? So I was walking down the street the other day at night. I don't know, it was 9 o'clock at night. There was a guy peeing on the street. And I was preparing for this lecture, and I thought, wow, if I was a female prairie vole, that would be my Prince Charming right there, the guy peeing on the street. On the one hand, it makes things simple, right? Just whatever. But on the other hand, humans, it just points out the difference that humans are very different. So the question is, I'm telling you all these great stories and these very significant, hopefully you'll see that these are very convincing results in prairie voles, that in their system, where they have very, very strong pair bonds, oxytocin and vasopressin are playing very specific roles in at least this one aspect, partner preference. But what about humans? We're so much more complex. What can we do? So I'm just going to give you one simple experiment that was done, I think back in about 2000. Was it 2000? Yes. The neural basis of romantic love. And what they did is an fMRI experiment so they can look at brain activations in people that reported themselves to be in deep romantic love. So, soon after you form your relationship with somebody and you're in that deep romantic love situation, they recruited couples like this, and they scanned the females, and they showed them pictures either of their partner at the moment, the one they were deeply romantically in love with, or a friend that's the same age, that they've known the same amount of time, and they compared brain activations when seeing the romantic partner and the friend. And first, they looked at just ratings of feelings of love and sexual arousal for the loved partner and the friend. So yeah, a little bit of love for the friend, but much more love for the loved partner, and a small amount of such sexual arousal for the friend, and much larger for the loved partner. So this kind of behavioral assay matched up nicely, said that this is a nice comparison. So what they're going to do is look at activations for loved partner, looked at brain activations for friend, and subtract the activations for the friend from the activations from the loved partner, and see what is left. And that means that we're going to look at what brain area is left when you just look at pure activation in response to the loved partner. And so, just very simply, we have a number of activations, and some of these areas will be quite familiar to you. We haven't talked about the medial insula, anterior cingulate cortex. So can you see this? This is a mid-sagittal view. This little yellow blob is part of the anterior cingulate cortex. This was the original part of that limbic system. It turns out that the anterior cingulate cortex is involved in not so much emotions, and has been implicated in these kind of love feelings, anterior cingulate cortex. Interesting, caudate, putamen, and globus pallidus. We talked about these areas in Parkinson's disease. But what we didn't mention in Parkinson's disease is these same areas are also very strongly implicated in reward. That is, using reward to teach you what is good and what is positive and what is negative. So these areas very strongly implicated in reward are activated in the loved partner when the female is looking at a picture of her loved partner. Hippocampus gets activated. Hypothalamus, again, part of the limbic system, originally identified in sham rage. But hypothalamus, as part of this oxytocin and vasopressin kind of response, and VTA is ventral tegmental area. It is the area that's supplying dopamine to the caudate, putamen, and globus pallidus, and is another area very strongly implicated in reward. So those are the activations. These are areas that get activated when you're looking at a picture of somebody that is that you're very much in love with. Now you can ask, well, what about the correspondence of these areas? Do they have vasopressin and oxytocin receptors? 2 points here. The first point is vasopressin and oxytocin receptors are not anywhere near the same locations in the human as they are in the prairie vole. We do not have our oxytocin and vasopressin receptors in the same places as prairie voles. But what we do have them in are places that also, many places that also get activated when you're looking at this picture of somebody that you're very much in love with. Lots of the receptors in humans in caudate, putamen, globus pallidus, hippocampus, hypothalamus, and VTA. So these studies in humans are still at their very, very beginning stages. But hopefully you'll be able to see how we can use clues from these animal model systems to start to ask questions. Not everything is going to be identical. The location of these receptors are not identical. But these studies and others have implicated vasopressin and oxytocin in the response, the love response, or responses to loved people. They've studied parent-child bonds as well, and gives us a great leg up to start to look at even higher levels of cognitive function, including the neurobiology of love. So I want to spend the next little bit of time just reviewing what we've gone over in this whole semester. I can't believe it's over. But final thoughts, I just want to go back to the 3 kind of goals that I started out with way back in September. And those goals for you for this class were to provide a little bit of inspiration, that something in this class would inspire you, catch your interest, hopefully from the cellular all the way up to the neurobiology of love. Something would be inspirational to you, to be very, very interesting, something you hadn't thought about before. Interaction, to allow you and I as a teacher to interact a little bit more, but more importantly, to inspire a little bit more interaction with you and the media out there, because hopefully, as you've appreciated, there's so much neuroscience in the media. Just a few days ago, the whole story, did anybody see the story about the hockey player that recently died? He was a very, very violent guy, and they went through a whole series, 3 days. Started on the front page of the New York Times, but it ended up in the sports section, talking about his behavior, which was kind of very, very aggressive. He was, what was it called? He's like a fighter. What's it called when you're a hockey player? What? Enforcer, thank you. He was an enforcer on a hockey team, which means that he was just the guy that fought a lot. And he ended up with severe brain damage, and they looked at his brain, and the brain damage came from all the fights and all the multiple concussions that he incurred over his, I think he was only in his late 20s, right? Was he in his 20s when he died? Do you remember? Does anybody know? 28, yeah. So, so much to understand in the media. So hopefully some of the data that we've gone over gives you a better and a deeper appreciation of that. And again, the third is just appreciation, not just of the media, but so many other things. I mean, we started with cells and the basic structure and synapses and neurotransmitters and hormones that are working at, in these beautifully drawn cells by Camillo Golgi. This is a picture I showed you in the first lecture. We went on to look at sensory systems, focusing on somatosensation, vision, and hearing, and all of those pathways, those many pathways and details about transduction all the way up to the cortical areas that process this information. And what do we do with that sensory information? But we can act on it. Our motor output and our understanding of how the brain and primary motor cortex eventually goes out and can control all the muscles in our body through these kind of beautifully designed motor output structures. And finally, using both sensory, motor, and cellular understanding of the brain to then move on to higher level understandings of cognitive function in cognitive functions in people, famous case HM, and not only in understanding what damage to this specific part of his brain that was damaged, what that does, causing a selective anterograde memory impairment for declarative memory, but also eventually trying to understand what are the cellular underpinnings. Why can the hippocampus do this for us? Is it because of LTP? Is it because of the habituation, that the same molecules that are involved in habituation? Lots of interesting questions there. And then all the way up to even what some might consider even higher order cognitive functions, like social and affiliative behaviors. But I just wanted to really end with just reminding you of what we started out with, which is how neuroscience affects all of us and everyday lives in ways that you might not expect. So I told you about the biography of this artist, Chuck Close, and I told you, and I kind of advertised to you, that we'd be talking a lot about lots of the elements in his life. Vision and the abnormalities of vision that he had. He had prosopagnosia, and you talked about, you learned about those ventral temporal areas, which when damaged causes prosopagnosia. I don't know whether Chuck Close has ever gotten an MRI, but that would be the prediction for why he has continued a very difficult time understanding and recognizing people based on their faces alone. But clearly, that was one brain difference that he had. Clearly, amazingly talented artist that went through these phases using this disability that he had to create this amazing, very striking, very kind of evocative art. They evolved even more after he had his, what I call the big event. So he had a spinal aneurysm, and he's in a wheelchair now, yet still able to paint. And so we've learned not only about the visual system, brain areas involved in prosopagnosia, but the spinal cord areas that are involved in his spinal aneurysm that causes his legs not to have motor function, but he still has pretty good mobility in his hands and his upper neck. So you should be able to know what spinal level his deficit came at. And then his later work, which has been described as much more emotional, much more warm, as he's evolved these faces from very, very stark and realistic representations. This is all done with blowguns, even though it looks like a photograph, to very abstract paintings in color now. And the brain areas that might allow us to appreciate that change from stark to warm and to evoke different emotions in us. We've talked about the amygdala in fear emotions, but frontal cortex in other kinds of emotions, and particularly positive emotions.